Thank you, Warner, for that wonderful introduction. I'm going to tell you about some of our work on investigating the mechanism of latent HIV infection, and I'll give you some background, some data that's recently published, and some data that's never been published. But just a word of apology, I'm getting over a cold, and so if I lose my voice or I clear my throat or need a drink, that's why. There you go. So just to start with a little bit of background, it's been known for about 20 years that HIV can establish a latent infection in CD4 positive T cells. These cells don't produce virus constitutively, but they can be induced under certain circumstances, especially by T cell activation, to produce virus that can infect other cells. The way this was discovered was they took patients who were suppressed on therapy, took CD4 T cells from the blood, and find that if you culture them in the lab, they don't produce any virus. But if you activate them, they can produce virus that can then infect uninfected donor cells, co-culture or QVOA assays. It's been known that these latent infected cells have a very long half-life on ART, over 6 to 44 months or longer, and they're widely regarded as the main reason that we can't cure HIV. In addition, it's thought that these cells are continuously reactivating in vivo, and that may contribute to immune activation, inflammation, and some of the organ damage that we see even in patients on suppressive ART. However, there's extreme challenges to studying HIV latent infection. Number one, these cells are extremely rare, 1 in a million CD4 T cells, but more importantly, there's no way to sort out latently infected cells from patients. In fact, we don't even have any way to sort out infected from uninfected cells, let alone those with infectious proviruses or those that can be induced. And there's multiple models of latency, but unfortunately, there's no agreement on a gold standard. If you use them, they show varying responses to drugs designed to reverse latency, and these models have largely failed to predict the results of clinical trials. So, as I said, despite 20 years of research, it's very unclear what mechanisms govern latent HIV infection in vivo. And it's not that we don't know any mechanisms. In fact, as far as I can tell, there's been at least a dozen mechanisms that have been implicated in HIV latency. And here I show you a schematic of the HIV DNA or provirus, some of the transcripts, and then some of the mechanisms that have been implicated in latency. But while some of these studies have used blood cells, most of these studies have been done in various in vitro models, and most studies only investigate one mechanism at a time and then say, hey, this mechanism is important. So one thing we really realize is it's very important to figure out, well, to what degree, if any, are these various mechanisms operating in patients? Unfortunately, it's very, very hard to figure this out. But one of the realizations we had was that these mechanisms will produce different types of transcripts. For example, if you have lack of, oops, sorry, didn't mean to do that. If you don't have initiation of HIV transcription, you'll have HIV DNA, but you won't have any HIV RNA, except for one particular mechanism called transcriptional interference, which is where you have transcription from a neighboring human gene that continues into the HIV DNA, prevents binding of factors to the HIV promoter, and blocks HIV initiation. This particular mechanism, you'll get HIV transcripts that actually start upstream of the usual transcription start site in the TAR region, and will give you read-through transcripts. But in general, if you don't have initiation, you're going to get HIV DNA, but no RNA. In contrast, you have mechanisms that block the elongation of HIV transcription, and these are going to give you short HIV RNAs, but not elongated fragments. And then there's some mechanisms that operate after transcription has been completed, and they should give you full-length polyadenylated transcripts, in some cases multiply spliced transcripts. So what we realize is if we look at all these different types of transcripts, and especially we're able to quantify them and look at the ratios, you can figure out at what steps HIV transcription is being blocked and to what degree. And so this slide summarizes our general approach to do this, which I'll call transcription profiling. And again, at the top, I have the organization of the HIV DNA or genome, some of the transcripts, and then some of the assays that we measure for different HIV RNA regions. For example, we have an assay for the TAR region, which is the very first region transcribed in HIV and is present in all transcripts. So this can tell you about initiation or total HIV transcription. We have another assay that, sorry, I keep doing this. We have another assay that goes beyond the long terminal repeat region to just before GAG that is downstream and measures elongated transcripts. We adapted another assay that can pick up polyadenylated or completed transcripts. We developed another assay for multiply spliced TAT-REV transcripts. And then later on, we realized that we needed additional transcripts, and so we developed assays for the Pol region, which is sort of in the middle of the virus and present only in unspliced transcripts, and another assay for the Neph region, which is present at the very 3' end and should be in all transcripts. And for those of you who don't study HIV transcription, I know this is probably very confusing, but a very simplistic way to think of this is that we develop assays for different HIV RNA regions that are sort of 5' to 3', and also represent different stages of HIV transcription or different blocks to HIV transcription. And our general approach has been that we take blood or tissue cells from patients usually, or sometimes models. We simultaneously extract the DNA and the RNA and measure the HIV DNA and housekeeping genes for cell numbers, and then we look at different HIV RNA transcripts. We use droplet digital PCR because it gives you absolute numbers and it's relatively free of inhibition due to sequence mismatches, which is important. And then we developed many special modifications to try to eliminate bias towards any one transcript, which is very important in trying to understand these. And we've done validations to show that the efficiency of these assays is all very similar. So our objectives were to use this panel of HIV RNA assays to apply to cells, especially from ART-treated patients, in order to determine the degree to which different mechanisms are inhibiting HIV transcription in the body, and also the degree to which these are reversed by physiologic T cell activation outside of the body, which is how we define latency, as well as the responses to different latency reversing agents. And when we started this, we thought that mostly what we would see is that latency is due to lack of initiation of HIV transcription, because this is sort of the prevailing model in the field. So this next slide shows some published data. What we did is we took blood cells from, I think it's something like 15 or more ART-suppressed patients, isolated the CD4 T cells, and then in each CD4 T cell, we measured each of our 5 main transcripts. And so on the y-axis here, I'm showing you the copies of each HIV RNA per microgram RNA. It's a measure of HIV transcription relative to cellular transcription, and a microgram RNA is worth about 1 million cells worth. And note that's on a log scale. And on the x-axis, I show you our assays for different HIV RNA regions, and each color is a different patient, and the bar represents a median. And you can see that when we look at read-through transcripts, which are suggestive of transcriptional interference, we find them in all patients. However, the levels are 100 times lower than total or initiated transcripts, suggesting that although this transcriptional interference is going on, it plays a relatively modest role. Our levels of initiated transcripts were in the tens of thousands, which is much higher than most people detect, probably because we have a special assay that can detect short transcripts. And this is suggesting there's a lot more initiation of HIV transcription than was previously recognized. Interestingly, if you look at initiated transcripts, the levels are 10 times higher than 5' elongated transcripts, suggesting you have a big block to elongation of HIV transcription. And very much to our surprise, when we look at 5' elongated transcripts, those levels were 10 times higher than polyadenylated transcripts, suggesting that even when you elongate past the 5' LTR, a lot of those transcripts never make it down to the 3' end and or never get polyadenylated. You never get completion of transcription. There's a block to completion. And finally, when we looked at polyadenylated transcripts, those were more than 10 times higher than levels of multiply spliced transcripts, suggesting that a lot of completed transcripts just never get spliced, that there's a block to splicing. So to summarize, it looks like at baseline in cells straight out of the patients, there's very strong blocks to elongation and completion and multiple splicing. We were particularly interested in this difference between the 5' elongated transcripts and the completed transcripts. And what is going on here is that they have 5' elongation but just never get down to the 3' end, or is it that they get down to the 3' end and don't get polyadenylated? And to sort of figure this out, we had to develop new assays for Pol, which is sort of in the middle, and then Neph is at the 3' end. And so we measured all 7 assays in here. I think it's 6 or 7 more patients. And what we saw is a gradient from initiated transcripts to 5' elongated to sort of mid-transcribed to distal transcribed to polyadenylated. And this suggests that this block to completion that I was describing is probably a combination of lack of distal elongation as well as maybe lack of polyadenylation, because in a lot of patients we see a difference between the distally transcribed NEF and the polyadenylated transcripts. Since levels of these transcripts can be influenced by the stability of the RNAs as well as the rate of production, we also wanted to look at the stability of the RNAs, and we did this by taking blood cells from ART-suppressed patients and then incubating the lab with compounds that block all transcription and measuring the decay of our different HIV transcripts. Here I show you the decay relative to time 0, which is 100% of all our HIV transcripts over time. And basically what we found is that the half-lives of these transcripts are all quite similar, suggesting the differences in levels that we're seeing can't be explained by differences in stability or degradation. Now, I've described what looks like these blocks to transcription at different stages at baseline, but an equally important question is, well, to what degree are they reversed by T-cell activation, which is supposed to be the stimulus that reverses latency. And so to do this, we took another 12 patients. We took CD4 T cells straight out of the patients and froze them for analysis, and then we also took another aliquot and activated it for 1 or 2 days with ARVs to block new infection. And then in both the cells from day 0, which are straight out of the patient, and the cells from activation day 2, we measured our 5 main transcripts. Here, I know there's a lot of plots, but I'm basically showing you the data for each transcript separately. The y-axis is copies per microgram RNA, which is basically looking at the degree of HIV transcription relative to average cellular transcription. And then I have the unactivated day 0 and the activated day 2 with different colors for each patient. And what we found is that activation didn't really have much effect on read-through transcripts, which are pretty similar between day 0 or 2. To our surprise, activation did very little to increase initiated HIV transcripts, at least by this measure of normalization. In contrast, we saw an increase in 5' elongated transcripts of almost 2-fold, an even greater increase in polyadenylated transcripts of about 11-fold, and an even greater increase in multiply spliced transcripts of about almost 30-fold. And what this is suggesting that you're not just having more initiation of transcription, you're having sequentially greater increases at the stages of elongation and polyadenylation and multiple splicing, suggesting that really the main reversible blocks to HIV transcription are not transcriptional interference as measured by read-through transcripts or lack of initiation, but really it's these blocks to elongation and completion and splicing. Another way of looking at this data is instead of looking at the absolute increase in each transcript, we can use the ratios of RNA regions before and after each block to figure out the proportion of transcripts that are blocked at each stage. For example, if you look at elongated transcripts and you compare them to initiated transcripts, you can use that ratio to figure out how much elongation there is, or the inverse of it to figure out how much of a block there is to elongation. So what I'm showing here is the proportion of RNA that's blocked at each stage of HIV transcription, with 1 being 100% block in the cells straight out of the patient, that's at day 0, and the activated cells at day 2 for each of our stages of transcription or blocks. What you can see is that at day 0, most of the transcripts are blocked at the stages of elongation and completion and splicing, but if we activate the cells, we see much less of transcriptional block at each of these stages, especially at the block to completion, which seems to be the most reversed by activation if you use this measure. Now, everything I've shown you so far has been by looking at HIV RNA levels in millions of cells, in bulk cells, and that's very informative, but at the same time, it can't tell you about the distribution of these transcripts and these blocks. For example, are all these transcripts in one cell? Do these blocks happen in every cell? And so in order to get an idea of this, we took a very different approach. What we did is, again, we took cells from ART-suppressed patients, but now we did serial replicate dilutions, both of the cells straight out of the patient and the activated cells. And at in each replicate, we measured all of our transcripts, which is a ton of work, and then we used the frequency of replicates that are the proportion of replicates that are positive to figure out the frequency of cells that have each of these transcripts. And since we can measure HIV DNA, we can also look at the frequency of infected cells that have each of these transcripts. So on the left, what I'm showing you is the frequency of infected cells that have each of these transcripts, with 1 being 100%, 10 to the minus 1 being 1/10. And somewhat to our surprise, what we found is the relative order of these cell frequencies is exactly similar to the order of the levels of the HIV transcripts per million cells, suggesting that these transcripts and these blocks are happening in most cells. In fact, if you look at the frequency of infected cells that have initiated transcripts, it's almost 100%, suggesting that almost all infected cells have initiation of HIV transcription. We can use the frequency of cells that have each transcript to figure out the frequency of cells that have blocks. For example, if you look at the frequency of cells that have initiated transcripts and the frequency of cells that have elongated transcripts, you can use the ratio to figure out what frequency of cells have a block to elongation. So on the right side, this is what I'm showing. The y-axis shows the proportion of infected cells that have blocks at each stage, with 1 being 100%. On the x-axis are the different stages, and day 0 and day 2 are the unstimulated activated. And you can see that in cells straight out of the patient, most cells have these blocks to elongation and completion and splicing. And when we activate them, that proportion decreases dramatically, suggesting again that these transcriptional blocks are operating in most infected cells in patients. I've shown you what happens when you activate these cells, at least ex vivo, but an equally important question is, we have all these drugs that are supposed to reverse latency reversing agents, or LRAs, but to what degree do these reverse the blocks to HIV transcription that we think are the mechanisms of latency? And so this is some unpublished data, and what we did here is we took peripheral blood mononuclear cells from, I think it's 11 patients. We took at least 5 million cells in replicate aliquots and cultured them for a day with either DMSL, that's in gray, that's our negative control, or antibodies to C3 and 28 to activate them, that's our positive control in red, or different putative latency reversing agents in different colors. And these, we chose things that work by different mechanisms. So here the y-axis is showing you the copies per microgram RNA of each HIV transcript, roughly the copies per million cells. And on the x-axis are each of our main different HIV transcripts separated by dotted lines. And then the horizontal dotted line at the bottom is our level of detection. And this is a lot of data and hard to take in at first glance. But if, for example, you look at read-through transcripts, what you can see is that none of the latency reversing agents really seem to increase read-through transcripts much, at least in a way that's statistically significant. If you look at HIV transcriptional initiation as measured by the TAR region, some of these LRAs do increase initiation. For example, disulfiram in green increases initiated transcripts. Our 2 HDAC inhibitors in blue increase HIV transcriptional initiation, and ingenol, which is a protein kinase C agonist, increases initiation. However, if you look at elongated transcripts, the number of LRAs that increase it is smaller. You still see disulfiram increases elongation. The HDAC inhibitors increase elongation, but then the other drugs do not. If you look at polyadenylated HIV transcripts, it's an even smaller subset of LRAs. It's really only romidepsin that increases polyadenylated transcripts. And then when you look at multiply spliced HIV transcripts, none of the LRAs really cause any significant increase in multiply spliced transcripts. The only thing that increases it is activation. So this data suggests that LRAs are really exerting different effects on these different blocks to HIV transcription, and also that the mechanisms underlying these different blocks may be very different. And I think it's particularly compelling to note that none of these LRAs really induced multiply spliced HIV transcripts. And we think that this is really essential for an agent to reverse latencies. It's got to affect this block to splicing and increase multiply spliced HIV RNA. That data was all done using pre-BMCs from patients that we treated in vitro, but an even more important question is what happens in vivo. And so we have a grant to look at a number of clinical trials where patients are getting latency reversing agents. So in this particular slide, we looked at the REDUCT Part B clinical trial where patients had vaccination followed by romidepsin. There were 17 patients in the trial, but we had only access to 11. And in these 11 patients, what we did is we measured each of our HIV transcripts before romidepsin, that's visit 1, and at 2 visits right after getting romidepsin, those are visits 10B and 11B. And each graph here shows a separate HIV transcript. Each dot is a separate patient with the y-axis on a log scale, the bars representing the median, and when we couldn't detect it, it's a solid circle. But looking at these graphs, what you can see is that in vivo, what romidepsin seemed to do, at least in this patient, is it increased read-through transcripts, it increased initiated HIV transcripts, elongated HIV transcripts, and even polyadenylated transcripts, but it didn't seem to do anything, as far as we could tell, to multiply spliced HIV transcripts. And when we took a closer look, looking at ratios, what we saw is that initiated transcripts, the absolute increase was much greater than read-through, suggesting you really are getting specific HIV transcriptional initiation. When you look at the ratio of elongated to initiated transcripts, it goes up even more, suggesting that really the main effect of romidepsin is increasing elongation. In contrast, when you look at the ratio of polyadenylated to elongated transcripts, it actually goes down, suggesting there's no specific effect on completion, and the increase in polyadenylated transcripts we're seeing just reflects more initiation and elongation. And then, as we said, we don't see any change in multiple splicing. And we think that this data is really compelling because it's really suggesting that romidepsin is not specifically increasing production of completed or multiply spliced HIV transcripts. And we think that these are essential for cell killing because it's probably the polyadenylated transcripts that get exported from the nucleus, that are stable, that get translated and made into proteins, that get cleaved to peptides and presented, and also trigger intracellular defenses. So we think this is probably why romidepsin and a lot of latency reversing agents are not really leading to a death of infected or latent infected cells in vivo, because they're not really producing these polyadenylated multi-spliced transcripts. They're not overcoming these blocks, and also probably not really effectively reversing latency. We had some preliminary data to suggest that things might be very different in the tissues, especially the gut, which is where most HIV-infected cells reside, and that latency might be very different in the gut. And so to take a look at this, what we did is we took ART-suppressed patients, we got blood and gut samples from the same patients, and then we FACS sorted out the CD4 positive T cells from both blood and gut. We measured HIV DNA and RNA. On the y-axis here, I'm showing you the ratio of HIV RNA to DNA. That basically corrects for differences in infection frequency. In addition, it expresses the average level of each HIV transcript per provirus. So the y-axis is the average level of each HIV transcript per provirus. On the x-axis, I have a different HIV transcripts, and I always list blood and then rectum, and the different transcripts are separated by dotted lines. And what you can see here is if you look at HIV transcription initiation as measured by TAR transcripts per provirus, in the gut, there's more than 10 times less initiation of HIV transcription, which is very, very surprising to us because in the gut, more cells are activated, and activation is supposed to be what reverses latency and increases transcription. And so it's very paradoxical that you're getting a lot less initiation of HIV transcription in the gut. When we look at 5' elongated transcripts, it's very different. There's very little difference between blood and gut. And in fact, there's little difference in the gut between elongated and initiated transcripts, suggesting there really may not be much of a block to elongation in gut CD4 T cells. At the same time, I'll say that we did the similar studies in intact biopsies, and there we do see a difference between initiated and elongated transcripts in the gut. So it may depend on what type of cells you're looking at. However, in both types of gut samples, if you look at elongated HIV transcripts, the levels are much greater than polyadenylated transcripts, which are much greater than multiply spliced transcripts, suggesting that in the gut, as in the blood, you have these profound blocks to completion of HIV transcription and splicing. And based on this data, we wondered whether latency reversing agents would exert different effects in the blood and gut. And so this is some unpublished data. And basically what we did is we took blood cells, PBMCs, as well as total dissociated gut cells from ART-suppressed patients. We incubated at least 5 million for a day with either DMSO as our negative control in gray, or anti-CD328 as our positive control in red, or different latency reversing agents and ARVs. And we looked to see to what extent these LRAs induced HIV transcription in the blood and gut. And what we saw is that there's similarities as well as differences. Notably, in both the blood and gut, you see this mountain pattern. It's the same pattern, although there's less overall transcription in the gut. One of the interesting things we've found is that in the gut, you can induce HIV transcription with activation. This has never been shown before. It's not necessarily straightforward. It suggests that even though there's more activated cells in the gut and they have less HIV transcription, if you give them the right activating stimuli, you can induce all your HIV transcripts, suggesting that there is an inducible reservoir in the gut, which is probably latent, although we haven't proven it, and this may contribute to rebound. And then as far as particular LRAs, we saw similarities as well as differences between blood and gut. For example, romidepsin, which is an HDAP inhibitor, increased initiation and elongation and polyadenylation in both sites, but the increase in initiation was a lot smaller in the gut compared to the blood. Disulfiram, which is shown in green, increased initiated and elongated HIV transcripts in the blood, but we couldn't detect any effect in the gut. In contrast, ingenol mabutate, which is an agonist of protein kinase C, seemed to have more effect on the gut than the blood, particularly as far as increasing polyadenylated transcripts. And again, none of these LRAs induced multiply spliced transcripts in either the blood or the gut. And so this slide just summarizes a model of some of the mechanisms that may be different between blood and gut. In both sites, we probably have blocks to completion and to multiple splicing, but in the gut, we have a much stronger block to initiation of HIV transcription. We've also started to look at primary cell models of latency. There's multiple models that have been developed that differ in the viruses, whether they have reporters, whether they're replication competent, as well as the cells that are infected and how you induce latency. And really we wondered, well, how do these models compare to each other and the patient cells in the mechanisms of HIV latency? And so to look at this, we looked at 3 models, pre-primary cell models from Eric Verdin's lab, which is in orange, Werner Green's lab in green, and the lab of Alberto Bosque in purple, as well as cells from patients that were unstimulated or activated. And in each case, we had latent cells from the model, and we had productive or activated cells from the model. And in all of those, we measured our levels of our different HIV transcripts. And so here, the y-axis shows the levels of each HIV transcripts per HIV DNA. So that corrects for differences in infection frequency, gives you the HIV transcripts per provirus. The x-axis of our different transcripts, and it's a lot of data, but basically what we saw is that all these models are different from each other and from patients. There are very interesting differences between the models. For example, in the model from Werner Green's lab, when we activated the cells, we saw no difference in initiated or elongated or polyadenylated HIV transcripts, and the only thing we saw is an increase in multiple splicing, suggesting that in this model, the main thing that's maintaining latency is a block to multiple splicing. In contrast, when we looked at Eric Verdin's model and Alberto Bosque's model, we saw that the activated cells or productive cells had more initiation as well as an even greater increase in multiple splicing. And indeed, one of the most interesting things to come out of these models is that in all of these models, there's a reversible block to multiple splicing, suggesting that this really may be a key feature of HIV latency in primary cells. In order to try to figure out what cellular factors might be modulating these different blocks to HIV transcription and latency, we've turned to single-cell studies from both primary cell models and patients. In this slide, I'm showing you some data from Eric Verdin's model. And just to remind you that this model has 2 reporter proteins. One is constitutively expressed. The other is only expressed if there's transcription from the HIV promoter. When both are expressed, it's a marker of productive cells. When it's just the constitutive one, it's latent. And then you can sort out one that has neither marker or cells that haven't been exposed to virus. And here what we did is we took all these populations from this primary cell model, and we used index sorting to sort out one cell in a well into lysis buffer. We've developed a special workflow that will allow us to screen whether each well has an infected cell and even to get sequence information from it. And then we take each single cell's cDNA and use the Biomark platform to separate it using microfluidics to do 96 separate assays. So what I'm showing you here is results from Eric Verdin's model. Each row is a different assay. Each column is a different cell. And at the top, I've given different symbols for the different types of different phenotypes here. Our panel of 96 assays includes 8 different HIV regions, including multiply spliced and polyadenylated HIV transcripts in the blue box here, which we can detect, as well as different assays that detect HIV DNA and RNA, including we adopted Bob Silicano's assays for intact proviruses, and you can see that these work very well. We also have cellular genes that are housekeeping transcripts and lots of genes that have been implicated in HIV infection or latency, and these work very well. And you can see that in this study, the productively infected cells from his model and the green triangles cluster very differently from the latently infected and the double negative cells. This is similar data from Alberto Bosch's model, which uses a wild-type virus, and you can see all our HIV transcripts down here that are working well. And we're using the same thing in other models and in patient cells. And then finally, this is the last slide. I'll just show you one of our other studies we're doing. One of the limitations of the work we've done so far is we can't tell which of our transcripts are made by infectious proviruses, but we have figured out a way to do this, which is that you can dilute proviruses down to the single provirus level, and we can still measure our different HIV transcripts and blocks, and at the same time you can sequence almost the entire provirus and determine whether it's intact, infectious, or not. And so in this grant, we're going to be looking at cells from patients, from different tissues, determining whether they're infectious or non-infectious proviruses, and looking at their transcription, as well as their integration site and chromatin accessibility. And so just to conclude, we think that the main reversible blocks to HIV expression in circulating cells are blocks to elongation and completion and multiple splicing. These blocks operate in most cells, and we think they're the mechanisms latency in the blood, although in the gut there may also be a much stronger block to initiation of HIV transcription. Some implications are that latency reversal probably requires agents or combinations that reverse all these blocks, especially the one to splicing. Different therapies may be required for different tissues. And if you're interested in latency silencing, there's multiple different steps at which you can try to silence HIV transcription. And gut cells that are activated but don't transcribe much virus may be a model for doing so. This slide summarizes some ongoing studies, but I'm out of time, so I'll just skip to the acknowledgements. I'd like to especially thank the people in my lab, former and current lab members in green, but especially Peggy, Sushama, and Sarah, who have generated most of this data. Joseph Wong, who's my mentor and now my close collaborator, and then my other collaborators, Ma, gave us most of the gut biopsies. Jeff and CAL did a lot of the sorting. Peter and Steve gave me a lot of the patients. Ole and Martin gave me samples from the Robertson study. Mauricio, Warner, Emily, Eric, Indra, and Alberto have given us cells from the primary cell models. And finally, I'd like to thank our funders, especially the NIH, NIDDK, and NIAID, and CFAR. And just to remind you, my first grant that I actually got was a CFAR grant, so I'm very much in debt to Paul and Warner, so thank you very much to both of you. And now, recently, there's 2 people in my lab who have grants from CFAR. So thank you very much, CFAR. I think it makes a huge difference in the lives of new investigators.
So, Steve, how do you propose to overcome this multiple splicing defect?
So one of the things we really need to know are what are the cellular factors that are upregulated or downregulated by activation that may modulate this. And we're actually, as you probably know, using your model where the block seems to be just a splicing to figure out what cellular factors involved. Once we do that, we can think about specific modulation of these cellular factors. But just to put it out there, there are actually molecules that have been developed that either inhibit or promote splicing. Some of them are in early studies in cancer therapies, and we're very interested in trying these in, for example, your primary cell model and in cells from patients to see what they can do.
But so could it be something as simple as a relative TAT and REV deficiency? That, in other words, you don't multiply splice, you don't get TAT or REV. And what about the potential for supplementing a good initiator of transcription with TAT and REV? I mean, there are toxicity concerns, I guess, but
I think TAT and REV are critical, and that's really a lot of what our data is showing, is that if you get a block to multiply splicing, you don't get the TAT-REV multiply spliced RNA that makes TAT protein. And if you could get that TAT protein, you could probably reverse latency. You probably can't give TAT itself because it's too toxic, but you can, I think people are trying to develop both agonists and inhibitors of TAT, and there's some like corticostatin A that seem to be very powerful for inhibiting TAT. And I think those are very good potential therapies for either latency silencing or latency reversal. In addition, you could try to develop molecules that modulate splicing.
Peter.
Hey, Steve, that was great. The work you're doing on the gut is really important. And I was struck by a comment that you made that when you looked at extracted cells, you did not see a difference between total and elongated transcripts. But when you looked at whole biopsies, you started to see the difference. And is that because there were fewer transcripts in the total transcripts when you looked at whole biopsies, or is it because there was more elongated transcripts, so when you looked at the whole biopsies? And then as a follow-up, does this give you any insights into the cellular mechanisms that might be contributing to transcriptional blocks in the gut?
That's a great question, Peter. I should just clarify, when we sort out the CD4 T cells from blood and gut, we don't see much of a block to elongation in the gut. However, if you use total digested cells or you use intact biopsies, you do see a block to elongation. Part of the explanation could be that you get different subsets of infected cells in the biopsies or total cells that are not CD4 T cells or were not captured by the sorting. When you sort, you actually get much higher levels of each transcript and more robust data, and that's good, but in a sense, it may not capture all the infected cells in there, and there could be changes induced by the sorting. And we've tried to look at this, and at least if you use, for example, PBMCs, it looks like the digestion itself and the staining and the sorting have small changes in HIV transcripts, but the pattern remains the same. So we don't think that's what's going on, but it's tough because the effect on PBMCs of the digestion may be different from the total cells. So I don't know whether it's a difference in the type of infected cells or the methods or even the patients, but we've now done a lot more studies where we look at intact biopsies or total cells, and you do see a block to elongation in the gut, for what it's worth.
Steve, thank you very much. Appreciate it.